Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. In this lecture series on special products coming from amino acids, I will be going over catecholamines here. Let's get into the details of catecholamines. Tyrosine is a precursor of three catecholamines that are there in our body. So, they are dopamine, norepinephrine and epinephrine. All dopamine, norepinephrine and epinephrine, they all coming from tyrosine as a precursor. Now let's get into a metabolic pathway to look into details, and then I will go over the points that I have written here. So tyrosine, it is hydroxylated first by tyrosine hydroxylase enzyme into L-DOPA. L-DOPA here is dihydroxyphenylalanine. So the tyrosine is hydroxylated into dihydroxyphenylalanine by adding an hydroxyl group, which is just besides the hydroxyl group that is already present originally in the tyrosine molecule. So a coenzyme for this reaction is tetrahydrobiopterin, which is converted into dihydrobiopterin. So BH4 is necessary for this conversion. Now once we get dihydroxyphenylalanine called L-DOPA, L-DOPA undergoes DOPA decarboxylase reaction where carbon dioxide is taken out of the reaction, and the coenzyme for this reaction is pyridoxal phosphate. Now let's see how a tyrosine in a neuronal tissue will be converted into catecholamines. Now very first reaction towards the formation of catecholamines is conversion of tyrosine into dihydroxyphenylalanine. That will be done by an enzyme tyrosine hydroxylase, and in this process the coenzyme is tetrahydrobiopterin, that is BH4, which is converted into BH2. Now dihydroxyphenylalanine, it has got two hydroxyl group adjacent to each other over the benzene ring. Because the structure of tyrosine, I am going to write that here, alpha carbon in the center, carboxyl group, H, amino group, primary amino group, then we have CH2, and then we have benzene ring, and to that benzene ring there is hydroxyl group attached. This is what is the structure of tyrosine. And now tyrosine hydroxylase enzyme, what it does, it is going to add one more hydroxyl group to this tyrosine, so just besides the original hydroxyl group. So and I am going to add one more hydroxyl group here. And now a benzene ring which is having a 2 hydroxyl group is now referred as dihydroxyphenylalanine. That is what is DOPA, DOPA, dihydroxyphenylalanine. And the point here is a phenyl ring having 2 hydroxyl group which is adjacent to each other. That type of ring we call it as catechol ring. This is a catechol ring. Catechol ring is a phenyl ring with 2 hydroxyl group adjacent to each other. So in dihydroxyphenylalanine, that's how you see it. Now what happens to this L-DOPA? L-DOPA in all dopaminergic neurons will be undergoing decarboxylase reaction to make dopamine. And dopamine is a catecholamine because it will have an amino group attached to it. Okay, because after decarboxylation, I am going to show that to you now. So this alpha carboxyl group, which I am boxing it here, this primary carboxylic group is removed as carbon dioxide in the decarboxylation process. And if that carboxyl group is removed, so the remaining group is an amino group there, and that's why it is a dopamine. It is dihydroxyphenylalanine with only amino group remaining. That is what is a dopamine. It is a catecholamine now. So dopamine with the amino group there is the catecholamine, and dopamine is the catecholamine, is one of the catecholamine. Okay, so that's what is the meaning of catecholamine, catechol ring with an amino group. And dopamine is a neurotransmitter. In all dopaminergic neurons, there is conversion of phenylalanine into tyrosine by phenylalanine hydroxylase, and then the conversion of tyrosine into DOPA by tyrosine hydroxylase, followed by conversion of DOPA into dopamine by DOPA decarboxylase. These are the three enzymes that are expressed in dopaminergic neurons, while phenylalanine is converted to tyrosine and tyrosine is followed by conversion to dopamine. So, what happens in other neurons like sympathetic neurons? Sympathetic neurons, their neurotransmitter, it is norepinephrine. So that means they are going to express one more additional enzyme. All sympathetic neurons, they are going to express an additional enzyme called dopamine beta hydroxylase. And this dopamine beta hydroxylase needs copper and vitamin C, and it's going to convert dopamine into norepinephrine. Now, in the adrenal medulla, where epinephrine is the one that is synthesized, and also in some of the central nervous system neurons, where epinephrine is a neurotransmitter, so what will happen is, so they are going to express an additional enzyme called phenylethylalamine N-methyltransferase. So this phenylethylalamine N-methyltransferase, it is going to convert norepinephrine into epinephrine upon methylation reaction, and the methyl group donor is 𝗠𝐵𝐴 So 𝗠𝐵𝐴 donates the methyl group, converts norepinephrine into epinephrine, while 𝗠𝐵𝐴 is converted to 𝗠𝐵𝐴. So this particular reaction is catalyzed by phenylethylalamine N-methyltransferase in the adrenal medulla. Epinephrine in the adrenal medulla, which is released into the circulation, will help in a response to stress, including its participating in gluconeogenesis, glycogen breakdown process. Note that a stress hormone, another stress hormone called cortisol. This cortisol, which is a powerful glucocorticoid, it will have a positive effect on, allosteric positive effect on also induction. It is going to induce the gene which is coding for phenylethylalamine N-methyltransferase. Thereby, more and more norepinephrine is converted into epinephrine in the adrenal medulla. So, cortisol, which is made in the adrenal cortex, will induce an enzyme in the adrenal medulla which makes norepinephrine into epinephrine, and thereby more and more epinephrine is synthesized in response to stress. So this is how cortisol and epinephrine, they work together to bring up glucose levels under fasting condition and under stressful situation by inducing glycogen degradation and also gluconeogenesis process. And also cortisol breaks proteins in the skeletal muscle and provide amino acids that are needed for gluconeogenesis. Remember, there are 18 out of 20 amino acids which are glucogenic. So cortisol will induce skeletal muscle breakdown to provide those glucogenic amino acids for gluconeogenesis in the liver. So this is how a synthesis of catecholamines will go on, and catecholamines are dopamine, norepinephrine and epinephrine. So depending on the number of enzymes that are expressed in the pathway, there are different neurons which will use these neurotransmitters. Dopaminergic neurons will use dopamine as a neurotransmitter because they will express only enzyme until DOPA decarboxylase. And then sympathetic neurons express additional enzyme, dopamine beta hydroxylase, and convert dopamine into norepinephrine. And epinephrine as a neurotransmitter, these neurons will express another additional enzyme, phenylethylalamine N-methyltransferase. So that's how it will go. So these are the special things, that is catecholamines, that will come from tyrosine. Although the pathway here, it shows conversion of tyrosine into DOPA, DOPA into dopaquinone, and dopaquinone into melanin, and that we are going to study later. So all that information I have written here in the slide. So tyrosine is a precursor for 3 catecholamines, which is dopamine, norepinephrine, epinephrine. And tyrosine hydroxylase needs tetrahydrobiopterin and makes L-DOPA. Dopa decarboxylase needs pyridoxal phosphate and makes dopamine. And dopamine beta hydroxylase needs copper and vitamin C and makes norepinephrine. And phenylethylamine N-methyltransferase needs SAM and makes epinephrine. And also note that to make SAM, that is 𝗂𝐵𝐷𝑖nosylmethionine, there is a requirement of vitamin B12 and folic acid. And out of all these enzymes, tyrosine hydroxylase is the regulated enzyme in the adrenal medulla. Now, dopamine in all dopaminergic neurons like basal ganglia, it is involved in voluntary movements, and depletion of dopamine has been noted in Parkinson's disease. That's an applied thing there. Norepinephrine, it is used in sympathetic neurons, and sympathetic neurons responds to fight or flight reactions and also all the sympathetic actions. Epinephrine, it is released from the adrenal medulla where it is synthesized, and it has more effect on the heart and lungs, and it has a response to stress. Now let's move on to see degradation of catecholamines. Catecholamines undergoes a sequential action done by catechol O-methyltransferase and monoamino oxidase enzyme. So with the sequential action of these two enzymes, dopamine will be converted into homovanillic acid, norepinephrine will be converted into vanillyl mandelic acid, and epinephrine will be converted into metanephrine and then vanillyl mandelic acid. So in order to differentiate between a norepinephrine and epinephrine metabolic end product, we will have to measure metanephrine, so that metanephrine differentiates the vanillyl mandelic acid that is coming from norepinephrine or epinephrine. So that means if a urine sample has both vanillyl mandelic acid and metanephrine, then it is the one that is coming from epinephrine. So that's about a special product that is coming from amino acids. More particularly in this lecture I was going over the special products coming from tyrosine, that is catecholamines. Thank you.